Okay, question 3bi. So we're using that information in the table and we're explaining a change in volume of urine over that period of the investigation. So the first thing to think of is if we're talking about changes in volume of urine, for example, if we've got not very much urine in the morning when you go for a wee, because you haven't been drinking lots, or when you're well hydrated in the day, you've got a higher volume of urine. All of that is controlled by the hormone ADH, antidiuretic hormone. So a little simplified summary here, which if you've got it in your head, fine, don't mean to patronise you, but if you haven't got it the right way round, then have something like this to help you remember. But if we've been drinking more water and there's more water in your blood to start with, that will lead to a decrease or less amount of ADH hormone produced, and we are going to have more urine. However, if you've had less water being drunk and less water in your blood, that means we need to try to retain as much water as possible, reabsorb as much water as possible from the nephron back into the blood. And what happens is, to cause that, is more ADH is released and secreted into the blood, and ADH makes the collecting duct insert the aquaporins, which we'll go into detail of later, and you end up having a lower volume of urine. So little summary if you want to, if your head's not straight with that kind of main facts yet. And then the marks for this question, I just stopped the video because I think this was probably the most challenging question to be able to explain in words. So in the first 30 minutes, that's after the people have had their big drink of water, or whatever it was in the question, we have got water is absorbed into the blood from the digestive system. So we've got a higher volume of water going into the blood. So we've got more water, so there's less ADH secreted. That's 1 mark after they've had their big drink. So because there is less ADH secreted, we're going to have less water reabsorbed. When we're looking at kind of 60 to 120 minutes, after we've had that excretion, well, when we've done lots of wee, basically, when we've had a large volume of urine has restored the balance of water potential in the blood, and we haven't got that extra water in the blood anymore because it's been excreted as urine, that's when we'll go back to ADH production occurring if it's needed, basically. So I think this one was the hardest one to answer. These bullet points that's a bullet point, sorry, as well. And it was a maximum of 3. These are the bullet points for the mark scheme for that one. Question below it, explain why the concentration of salt in the urine changed between 0 and 60 minutes. Concentration of salt in the urine changed because you've got the same amount of ions, but we've just got a larger volume of urine, so we've got more water diluting it. So the concentration would be lower. The actual amount of ions hasn't changed. The amount of salt there, grams of sodium and chloride ions is the same. It's just we've got more water, meaning it's more dilute. So that was the idea there. If you think about, if you do chemistry about different molar concentrations of acid, when the lab techs are preparing those different concentrations for you, they might have the same volume of acid in each beaker and dilute it with different amounts of water, different volumes of water, don't use the word amount, different volumes of water to get different concentrations of acid, like 0.5 molar hydrochloric acid, for example. So the idea was there that the amount of salt is not changing, or the mass of salt is not changing. The amount of ion stays the same. In a larger volume, it'll be more dilute, so it's a lower concentration. Question 4, explain how the formation of glycogen in liver cells leads to a lowering of blood glucose concentration. So it's telling you that glycogen is stored in the liver, and it's in the liver cells, and forming that in the liver cells leads to a low glucose concentration in the blood specifically, and it's asking you for detail on how. So this is one, actually, to be honest, that I think we could have gone into more detail of in lessons. So if you got this right, well done. I seem to remember on this one, possibly Jensen. There was a couple of really good answers, but well done, because that's on you. And listen to this explanation and make some notes on this bit. So first mark is for saying that glucose concentration in liver cells would fall or decrease when glycogen is being produced, and it would fall to a concentration below the concentration of glucose in the blood. If we are creating glycogen from joining monomers of glucose together, that means that the concentration of glucose in the liver cells where this is happening is going to decrease because it's being turned into glycogen. That's the first point. This will create a diffusion gradient between the liver cells and the concentration of the glucose in the blood. We'll have a higher concentration in the blood and now a lower concentration of glucose in the liver because it's been converted into glycogen. And as we know, in diffusion, substances will diffuse from high to low concentration. You know from work with Mrs. Holt last year that glucose diffuses by facilitated diffusion. So we had a bit of a made up term by a couple of people, diffuses facilitatedly is not kind of a phrase, so just stick to, like we're saying, water doesn't osmose, water moves by osmosis, substances move by facilitated diffusion using carrier proteins or channel proteins. And if you just put diffuses or just diffusion, that wasn't enough for a mark. You had to say facilitated diffusion and get that detail in. All about the detail, guys. Okay, it's my dog pitter-pattering that you can hear. Okay, now we're talking about gastric band surgery, aren't we? Gastric bypass surgery, sorry for this one. And we've got some standard deviation in the mix here. And we are going to answer some questions on groups of people who are diabetic and groups of people who do not have it yet. So first question, use data from the table. So it would be, I haven't put it here, but you do get a mark if you have put relevant data pulled out from the table, and we're suggesting why the scientists reached the conclusion that many of the patients who didn't have type 2 diabetes were at a high risk of developing it. So Mark was saying that insulin sensitivity was similar, so the numbers, and you could put those numbers, they were similar with those that were diabetic. There's also an overlap of the standard deviations, and their sensitivity did increase after the gastric bypass surgery. So any two of those would get you the marks for that. Shout out to Evie there. I'm not saying everyone else didn't have a good answer, but a particularly impressive there. And a shout out to Jensen for this next one. So does the data support the conclusion that the gastric bypass surgery cured many of the patients' diabetes, but some were not helped as much? And give reasons. So yes, the sensitivity does increase. We can see that from the numbers. It goes up, it gets higher after the gastric bypass surgery. But in the after figures, the standard deviation are large. So that means that some of the people, if you look at the actual standard deviation numbers, plus or minus numbers, some of the people showed little change, some of them would have showed no change, and some of them actually got worse. And then there's an extra mark for saying we can't compare this with normal, in inverted commas, sensitivity in non-diabetics and non-obese people. So those three were the marks that you could have got for this one, and really well done, Jensen, because I think you got 3 out of 3 for that. It was fab. Question number 5 was really well answered. Apart from the negative feedback, again, you kind of either got enough detail or you didn't. So I've written it again in a slightly different way, but it's the same 3 key points in the sentence. A change away from the normal will cause a response in the body which will reverse or reduce the effect of the original change, bringing levels back to normal. So they actually asked you that same question twice, really, but in a different context. The next 3 questions were really, really well answered. You just need to pay attention to when it says other. So if you put urine here, yeah, we know that urine is the main way that water is moved from the body. However, it tells you that water is removed from the body via the kidneys. That is making urine. So you could not get a mark for that, and that is why, because it said give 2 other ways. So just watch out for that in your exams. So sweating, or you could have put evaporation, but they're the same mark. Breathing out, water vapour, and feces as well. Lovely. Oh, here was my little diagram just to show negative feedback again. The response bit is either there and the response is there, a reverse to whichever change is happening. Hypothalamus, I think everybody got, and pituitary gland, everybody got. So really well done on that, guys. And turning over to the last 2 pages. So loads of shout outs for this one. So we had done it the lesson before. I'm willing to put money on the fact that Miss Griffiths probably went over with you the lesson before, because your answers were really detailed, or maybe it's down to you guys, not discrediting you, maybe you just went over this in detail, or maybe you just knew it for any other reason, but it was really well answered, especially by Ellie, well done, Evie, well done, Tanisha and Jensen, well done in particular. So we're using the information to explain how AD8 increases the movement of water from the lumen of the collecting duct into the blood. So the details we need to know is that when ADH is secreted and it gets to the collecting duct, I think it was Rowan or some people put extra detail here about it being a complementary shape and fitting into the receptors on the cells here that it's going to be a target organ for. This hormone will cause vesicles that contain aquaporins to be inserted into the wall of the collecting duct. Aquaporins, nice name because porins, pore, and aqua relating to water. So they are little channels that let water through. So 1 mark for saying that it inserts, ADH causes the insertion of aquaporins into the wall of the collecting duct. Another mark for saying that water goes through the aquaporins. Another mark for saying that it happens by osmosis, and then the water then goes into a capillary. Okay, question 6. You could have put your X here or here because it was quite hard to tell which one was highest, but that's what you got the mark for, showing one time when the insulin concentration in the blood would be highest. It's when you have a peak of glucose concentration in the blood, normally after eating a meal. Explaining why you chose that time would be because insulin lowers blood glucose levels when they are high, so when there's a peak you will get insulin production. And then describe 2 ways in which a similar graph could be drawn for a diabetic adult. So 2 key points. One is that generally the levels of glucose would generally always be higher. So general higher concentrations of glucose is your first marking point. And also you wouldn't get this dramatic then decrease in a really timely manner, because this is the negative feedback happening when you have got sensitivity to insulin and when you have got insulin production. So in a diabetic person, whether that's a type 1 that's not controlled or a type 2, you would have longer peaks, if you like. The peaks would not decrease, it would take a longer time to go down, and these levels of glucose after they've been elevated would remain high. That's your 2 marks for that one. Turning over onto your last page, we have got a bit of maths here. So we've got somebody who's got 20 grams of glucose in their blood. Their rate of energy use at rest was 480 kilojoules per hour. So what that means is 180 kilojoules of energy is used per hour. We'll come back to this because it's asking you to calculate the time. How long would this 20 grams of glucose in their blood last them? Well, we just try and help with, either you get it or you don't. And if you're struggling with the maths here, I've tried to make it kind of make sense as much as I can for you. So this is saying that 480 kilojoules of energy are used by this person in 1 hour. So to get the time taken, if there were 480 kilojoules, divided by 480, I'm trying to think about my thinking when I did this now, it would take an hour for 480 to be used. But we haven't got 480, we've got 20 grams, which gives us, where is it here, 16. So we haven't got 480, we've got 16 kilojoules for 1. But we haven't got 1 gram of glucose, we've got 20. So the first thing we need to do is to get the amount of energy released in, for 20 grams of glucose, we need to do 16 times 20. That gives us 320 kilojoules of energy released from this man's glucose store that he's got. Ignore Mr. Edmonds banging away in the kitchen there. Then we need to work out the time. So we are going to, similar to the idea and thinking here, we are going to divide by the rate for this man, because it would be different for people. So we divide by 480, which gives us 0.6 recurring hours. And you could get the full marks for that. So 0.7, if you rounded it up to 1 decimal place, would not be accurate enough. You'd need to say 0.67 hours, and that would be a tick, and that would be 2 out of 2. Or you could convert it into minutes. So to do that with 60 minutes in an hour, you times by 60, and the answer was 40 minutes. So if you got that, very well done. If you didn't, kind of pause this, go through, think, does that make sense? Come back to me if it doesn't. And our last question is talking about extra glucose being made available during the time in between meals to make sure that we've got a consistent supply of glucose in the blood, and we need to explain how. So this is my kind of thinking. If I was reading the question, I'll be like, extra glucose needs to be made available. It's when we haven't had a meal, that not necessarily no, but that we haven't had glucose in our meal, so there's low levels of glucose in our blood. So what's going to happen? So this is me brainstorming and kind of planning my answer before I write it, that I know that if there's low concentrations of blood glucose in the blood, then glucagon is our hormone we're talking about, and we're going to have these effects. Glucagon is going to have these effects on the body. So glycogen in the liver is going to be broken down and converted into glucose, which can be released into the blood to increase it back to normal levels. We are going to have gluconeogenesis possibly happening if we really need it. So glucose can be formed from amino acids and fats in the diet. So especially for people who aren't eating very much carbs, this would be one of the main responses of the body to try to get glucose available in the blood so it's available for respiration. And yeah, so both of those things release glucose into the blood. There's my little bullet point plan. Then I can just go and not worry about all my G words and what they mean. I've kind of got my head straight, done my little bullet point plan, and then I can write it. So we had 3 marks available. 1 mark was saying that glycogen is broken down to release glucose. That was 1 mark saying glycogen is broken down to release glucose. You've got another mark for the detail of saying that this occurs in the liver. Interestingly, you could get the mark for saying glycogenolysis, which is what we're talking about. Lysis is the breaking down of glycogen. But if you couldn't remember that word, or you thought, I'm not sure how to spell it, or I don't want to risk getting it wrong, you do get the marks just for describing what that is. Another mark for a key point in saying that glucagon is the hormone that is controlling this. And then a 3rd mark for saying another method of getting more glucose into the blood is gluconeogenesis, or again describing what that is. Mr. Edmonds, not helpful. He's got his AirPods in so he can't hear me. Glucose can be created from other substances in the diet like amino acids and fats, and that's called gluconeogenesis. Gluco is relating to glucose. Neo means new. Genesis is about producing something or creating something. So creation of new glucose from different molecules. And that was your 3 marks for the last question. So I just added on the bottom, and I wrote this on some people's or similar. If you maybe got a bit confused here, or you got your G words mixed up, do a bit of G word revision. So glycogen being the store of glucose and the polymer that's in the liver. Glycogenesis is creation of glycogen. So that's when we are forming bonds, glycosidic bonds between glucose, alpha glucose, and making glycogen to be stored in the liver. That's if we've got too much glucose in the blood and we want to decrease the amount of glucose in the blood, we can get it out by storing it as glycogen. Glycogenolysis we've talked about is the splitting. Lysis means splitting apart glycogen. Gluconeogenesis here, and glucagon, completely separate. This is the hormone which does the opposite to insulin. Okay guys, so please let me know if, hopefully that was useful. Let me know if there's any bits that you want to go over when we're back after half term. I can't promise to be on my emails all the time over this week, but if you have got something, do email and I'll try to get on them regularly just to help you if you need anything else coming your way. You've got physics and maths tutor to get some past exam questions, as well as the AQA website. You've got end of topic questions in the book as well, and please just let me know if we can provide you anything else that you need for this week. So keep up the good work. Well done for getting through to the end of this video, and I'll see you in a week's time.